welcome.
Okay, thank you. I'd like to start by thanking Speakers for Schools for giving me the chance to talk to you today about things that I find exciting, intriguing, interesting, and I like to have the opportunity to pass on some of this to as wide a group of people as I can. I hope today that I'm going to tell you some things that you already know, some things that you don't know, and some things that you find hard to believe until you see the evidence. So to start with, on the first slide, something you might not know, these are all animals that live in the Antarctic on the seabed. So the idea that the polar regions don't hold a lot of life is just wrong on the seabed in the Antarctic. And I'll come back to that later in the talk. So here we have the Antarctic. Some things you know. It's the world's coldest place, but it's also the world's highest, driest and windiest continent. It has ice covering over 99% of the continent. It's got 90% of the world's ice, 75% of the world's fresh water. So if you add up all the water in the Great Lakes, the rivers, the ice in the Arctic, there's more than 3 times as much fresh water here. And that's because if you look at the right-hand side of this slide, this big area here, which is bigger than Europe, on average, it's covered with a block of ice 2 miles thick, 3 kilometers thick. And that I often find hard to comprehend. It's twice the size of Australia and it sits over the South Pole. So it's a big place and it holds a huge amount of ice, which is a problem for us, even if parts of it start to melt. So what do we think about temperatures across the Earth? This is the standard view. And here we have, it's hot in the tropics, it gets cooler in the temperate regions, and it's cold in the poles. And that's true. But there's a lot more to this than just the immediate cold at the poles and hot at the tropics. So if you look at these data, these are slices through the Pacific Ocean and through the Atlantic Ocean. And the 2 types of data we've got here are the black data, which are the average temperature for a given place in those oceans as we go from the northern polar regions at 80 degrees north to the southern polar regions at 80 degrees south. We can see there's a monotonic increase in temperature up to about 30 degrees, because that's on the left-hand axis, these mean temperatures, the dark data, and then back down to about 0. So we can see here the polar regions are a little bit below 0 at the highest latitudes. The white data are the variation in temperature, and they're on the right-hand axis. And they go from 0 to about 15, maybe over 15 in the Atlantic where we are. So we can see, if we look at the variation, the poles have very low variation, close to 0, maybe a degree at the highest latitudes. And the variation in the sea is highest in the mid-latitudes because it's low in the tropics and low in the poles. And it's high in the temperate regions. So the polar regions are unusual for 2 reasons. One is they have the lowest temperatures on the planet. So temperatures are below 0 and the variation is close to 0 as well. So very little variation. And it's been like this for millions of years, maybe as much as 15 to 20 million years. And that's allowed some of the most unusual animals on the planet. And I would argue the most unique in terms of biodiversity animals on the planet to evolve in the Southern Ocean. So although the temperatures are constant, the seasonality is extreme. And this is a picture from a satellite on the left-hand side of the maximum extent of sea ice towards the end of winter and the minimum extent of sea ice towards the end of summer. The difference is between 10 and 15 million square kilometers of sea ice. And that doubles the size of the continent in the winter. And it has a massive effect on the sea underneath. It blocks out light. It reduces wind mixing. It makes the water column more stable. but it also then locks up nutrients from falling dust that then get released in a pulse at the start of the summer. It has a big effect on our climate as well, because this very large area of our ice reflects sunlight as the spring starts. And as we reduce that with warming, which seems to be happening quickly now, we reduce the amount of light reflected away from the planet. Antarctica is unusual in another way too. It is the most isolated continent on Earth. So if we look at these light blue areas, these are what's called continental shelf. And every continent has an area of relatively shallow water up to 200 meters deep around it, called the continental shelf. It's where most of our fisheries occur. It's where most of the useful things for humans occur. Antarctica is the only continent that doesn't have a continental shelf bridge with another continent. And that means animals can't walk from another continent to the Antarctic, which they can do with every other continent on Earth. So that isolation is more than anywhere else. Then the Southern Ocean circulates around the Antarctic, stopping things floating from other continents into Antarctica. So it's the most isolated ecosystem, set of ecosystems on the planet. And that means, again, things can evolve without being outcompeted by other species coming in from warmer places. So this is where I work, and I'm leaving for this station, Rothera Station on the Antarctic Peninsula, on the 28th of this month for a 2-month stint looking at biodiversity on the seabed. In the summertime, it looks like this. There are big pieces of ice in the sea, up to the size of very large blocks of flats. But we can get out in small boats. We can go to the sites where we need to do our experiments, take our measurements, or collect animals from or plants from to do experiments on. These are the most difficult diving conditions. So when the sea is full of broken ice and it moves around, it means that when you're on the seabed working, a piece of ice can come past and it can cause you big problems where you're working. It's also more difficult to get in and out of the sea, and if there's a problem, it's harder to retrieve a diver who's had a problem. In the winter time, the sea freezes, and where we are, it usually freezes to between 2 and 3 feet, so 0.6 to a meter thick. We take out our 5-foot chainsaw and we cut holes in the ice. We then put the divers into the holes. They're all roped so that they can get back to the hole if something goes wrong. And we carry out year-round diving operations. We're the only country to do long-term, consistent year-round diving operations. And it's part of our monitoring of the environment and monitoring of life and looking how seasonality affects life. I said early on that the biodiversity on the seabed in Antarctica was unusually great. So far, we've described over 8,000 species and named them. We know from the number of species we've not yet named, but we know are new, and from the number of species we've got from the amount of effort that we've put in, that there are at least 17 to 20,000 species living on the seabed in the Antarctic. This is a picture from 15 meters depth from an area where it's protected from ice, because iceberg scour removes all life from the seabed. But here we've got as much biodiversity and as much diversity of life and as much biomass on the seabed as you get anywhere outside of the coral reefs in the tropics. So very similar to what you would see off the west coast of Ireland here in terms of the diversity. Different species, very different attributes. But the biodiversity here is as high as it is in many other continents. So the idea that life is very poor and there's a paucity of life is just not true on the seabed in the Antarctic. Temperatures are low, and low temperatures affect biologies. It slows down growth, it slows down development, it slows metabolism, it slows how fast you can process meals, and that has massive consequences for the amount of energy that you can get in. It slows all the chemical reactions in the cells of the organisms that live there. And it's the same as doing chemistry practicals. If you want a reaction in a chemistry experiment to go faster, you heat it up. You put a Bunsen burner underneath it, and then the chemical reaction goes. If you cool everything down, all the rates go slower. So here we have data for oxygen consumption, and these data for a bivalve mollusk, so things like oysters, mussels, clams, and they're data from animals living in the tropics at 25 to 30 degrees, all the way down to the Antarctic and the Arctic at 0. And we can see that the metabolic rates, the respiration rates, the oxygen consumption are around 10 times slower than they are in the tropics. These are data for growth. And we can see that the growth rates are about an order of magnitude slower, maybe a bit more in the polar regions than they are in the tropics. And we can see that the fastest rates go up, the average rates go up, and the slowest rates actually do go up a little bit. And they all change by ×2 to ×3 for every 10 degrees of warming. Here we have data for development rates, and these are really slowed. So if we look at the time it takes for a brooding period for snails, and that's what we're looking at here, for snails that live in the tropics, in the warm temperate regions like the Mediterranean, in the cool temperate regions like where we are, in the subarctic and the subantarctic, and then in the Antarctic, we can see that the rates slow. And down to about 5 degrees, this graph looks like the previous two, where there's a general slowing everywhere. But then you get to very low temperatures, and suddenly there's a massive extra slowing of the development rate. So it's taking these animals, these snails that live in the Antarctic, 8 years and 10 years to go from laying fertilized eggs in their broods to them hatching out as little snails. In the tropics, it's a few days. Where we are, it might be a few weeks. In the subarctic and the subantarctic, it might be as much as 15 or 20 weeks. And then you get into the high polar regions where temperatures are 0 degrees or below, and these numbers go up to 100 and then 400 and 500 weeks. This is a really strong area of research in my group and in a few groups around the world as to why this is. And we think it's because the low temperatures, when you get close to 0, change the physics of the internal milieu, the internal environment of the cells. Water starts to freeze, it becomes sticky. And these animals have mechanisms that stop it freezing. But then that comes with extra problems with living in an environment where the water wants to stick together and starts to become sticky with other molecules. And this gives them real problems making proteins. And we know from other work that the rates of making proteins are 10 or more times slower than they are in warmer waters. So there's a big problem making proteins when your body is permanently at 0 degrees. These are data for different activities of animals doing different things: walking in limpets, swimming in scallops, drilling in predatory snails that drill through shells and squirt digestive juices into their prey, digest them and suck it back, walking in sea urchins, burrowing in clams, walking in starfish. And here what we've said is we've looked at speeds that this is done by species living at 10 to 15 degrees, and we've called that 1. And then we've looked at the rates in Antarctica compared to those rates at temperate species, and they're nearly all 2 to 10 times slower, 50% to 10% as fast. And that actually fits in with just a slowing by temperature. There are two things that are compensated for the low temperature: sustained swimming in fish, where they solve the problem by increasing the energy producing organelles, the mitochondria in their cells, by 2 to 3 times over fish in warmer waters, and burrowing in this clam. And this Latanula clam burrows as fast in the Antarctic as relatives that live in warm waters. And they do this just by increasing the size of the organ that they have for burying. So it's 3, 4, 5 times bigger in the Antarctic species than in the warmer species. So here we have some data for work we did on predation in a drilling snail. This snail, Trophon. It drills holes, it squirts digestive juices through, and it eats clams and brachiopods mostly. We wanted to know if these animals had feeding preferences and if the same individuals fed on the same things each time. In other words, they learn to prey type. So we did a 3-year experiment where we brought 100 of these animals back. We offered them a very wide range of potential food types in the tanks they were here, and we watched what they did. It took them 28 days to complete a meal. So you could pretty easily get data that they were feeding. And it takes 5 days in the UK and less than a day in the tropics. But in the 3-year experiment, they ate between 2/3 and 2.5 prey items a year. 2 animals ate nothing for 30 months and 1 animal ate nothing for 3 years. None of them had more than 8 meals in 3 years. And these 2 that ate nothing for 30 months laid broods after 17 and 22 months. And the 3 animals that ate virtually nothing were moving around the tank exactly the same speeds as other animals. What this tells you is that if your body is at low temperatures and you're living at low temperatures and things aren't perfect, you can just wait. And that's a massive advantage compared to species that are living at warm temperatures where their metabolic rates are so fast that they have to eat maybe every month, maybe every week, maybe every day, because they starve very quickly if they can't find food. So this ability to wait is an advantage of living at low temperatures. There are unique things.
Sorry, just wanted to let you know we're at 20 past now. So
okay. So we have giant species in Antarctica, sea spiders, sponges, and they're there because there's more oxygen. All the fish have antifreeze, and that allows them to live at 0 degrees in amongst ice. We have fish that don't have red blood cells. They're the only animals with backbones that don't have red blood cells. That's only allowed because the temperature's low, and they look very different. They have the ability to be almost invisible. Here we see blood from a red-blooded fish in Antarctica and blood from one of these fish without red blood cells. Their hearts are 5 times bigger. We have species that are biologically unique. Their capillaries are bigger than anywhere else. So, responding to change, our animals have very little ability in their biology to live above what they see, maybe 3 degrees, like the tropics. But animals living in between have 5 to 10 degrees of capacity to cope with warming with their biology. They all live a long time, and their generation times are long, which means they don't have a big ability to evolve an adaptation to a new environment. And their eggs are way bigger than they are in warm waters. So bigger eggs means fewer eggs. So again, fewer shots at evolving to the new conditions. So in summary, we have some massively unique adaptations that are so different that our animals are biologically different. They can't cope with warming. Their physiologies are very slow. They have problems making protein. And I don't see anybody pushing the let's save the Antarctic sea spider or the ice fish in the way that we do for elephants, lions, tigers, marmosets. But our animals are biologically more unique to biodiversity on the planet than any others. It's a great place to jump in the sea and go scuba diving and investigate the biodiversity. Let's hope climate change doesn't put the sun down on too many of our species. Thank you for listening.
Thank you, Lloyd. That was awesome. I definitely learn more than I ever have about polar marine life. Some really interesting stuff, and I think that was a great way to start. I'm going to crack on because we've got loads of questions. And we've had a question in from Jas, who was asked, where does the funding for these studies come from?
So it comes from 2 places. We get direct government funding to do things like monitoring. So we monitor the environment, and those environmental monitors are paid for directly by the government. The government also pays for the infrastructure. So it pays, you'll have seen we've had a new ship in the last 3 years, the Sir David Attenborough. That's paid for directly by government. And we've got stations in the Antarctic, 3 stations. They're paid for directly by government. Most of the science we do, we have to win grants for. So we apply for grants, and currently in the UK, success rates are about 10% in grant funding. So we spend a lot of time writing grants and not winning them. but then we do win grants for some of the research that we can do. And that's how we get the funding for most of the research we do, is through grants. The third way is the government gives money to fund the training of PhD students. So if we get a PhD student who's interested in our work from one of the universities, then they come with a small amount of funding and we can get support to do research through the PhD students.
Interesting. So once you have funding secured for a trip, how do you plan and organize these sorts of, remote field expeditions in these quite difficult environments?
It really depends where it is. So if it's in the deep Antarctic, then, and it's say on ice sheets and ice dynamics, you're taken in by aeroplanes, you have to be flown in. So the Antarctic Survey runs all of that infrastructure, and you will win money to do a specific piece of work, and you'll then interact with the operations teams in BAS to work out how to get you there, how to get your equipment there, and how to get it out. If you're working, say, in the Arctic, you'll have to get loads of permits from the indigenous peoples to work on their land. You'll have to get loads of permits from the Canadians or the Norwegians to work in their areas, as well as winning the grants to do the work. You'll probably have to have partners in the indigenous peoples, and you'll have to have the whole thing planned out probably 2 to 3 years in advance before you can get the infrastructure in place to be able to carry out the work. So it depends exactly where you are. So if I want to go and do a piece of work on our station, say I want to look at how fast the respiration rates are of some of the fish living near our station, that's a lot easier to organise because I don't need aeroplanes to carry me into the middle of the Antarctic. And our planning for a season is about 18 months to 2 years in advance. Fitting something in that is just on the station is usually less of a difficult thing than working out whether we've got the plane hours to take somebody onto Thwaites Glacier, as well as fitting in a season where somebody else has got funding to fly up the Antarctic Peninsula to look at Peninsula glaciers, for instance.
Very interesting. So obviously you just previously mentioned PhDs. What sort of education level background do you need to go into polar marine biology or just marine biology in general?
So there are lots of ways of being a marine biologist. The academic route, which you're talking about, you would routinely need to do a degree and then do a PhD in a marine subject, probably a master's in between as well. So you're looking at going to university for somewhere in the region of 7 or 8 years to get to the point whereby you're then trained as a marine biologist in your academic area, and you would then compete for jobs as a marine biologist. Being a marine biologist, though, is a kind of many splendid thing. And we have a whole range of support staff who are marine biologists, and they do a lot of the field work. So we have people who collect samples for us in the Antarctic, run the boats for us in the Antarctic, run the long-term monitoring in the Antarctic. And most of those have degrees. A lot of them don't have qualifications beyond degrees, but they've got a huge amount of experience of driving small boats, of running scuba diving operations, of being parts of expeditions elsewhere. So you can go on expeditions as volunteers with things like Operation Wallacea, or you can go on biodiversity expeditions in a whole range of places around the world. And lots of our people have volunteered for those, have done some marine biology, have spent a lot of time working as working marine biologists without being academics. So they don't write grants, they don't write papers, they don't become academics as marine biologists, but they have a life as working in the field as a marine biologist. There are people who become photographers and work in marine biology. You should look up Dougie Allen's career. He was one of our diving officers, and he ran our diving operation in the Antarctic for 2 years. He's now one of the world's most renowned underwater photographers and has taken video for Life in the Freezer and Blue Planet programs of killer whales going past and things like that. So he sees himself as a marine biologist, but he doesn't do experiments. He doesn't write papers. he goes out and he films. So there are lots of ways of getting into a marine biology career without being an academic. The most common view of a marine biologist is an academic, and then it's A levels, a degree, and then a master's, and then a PhD, and then compete for a job.
So next question then, thinking about, so a young person wants to go and take this step in their career path and go to university, or they're looking at their A levels, what sort of qualities, like personal qualities or really key skills, are needed for them to succeed in this as a career path?
So I often say to my students, when I talk to them as undergraduates doing their degrees, and we have career discussions, I often say to them, don't be a marine biologist if you want money, because the pay is really bad. Don't be a marine biologist if you want status in society, because accountants and lawyers and merchant bankers will always look down their nose at being a marine biologist. Be a marine biologist because you have got a genuine inquisitiveness that you can't switch off, that you really want to know how the natural world works. You are besotted by questions when you go home at night about why is life like this? Then be a biologist. And if it's why is life in the sea like this, and you can't stop asking those questions, then yeah, you've got to be a marine biologist, because the returns you get from understanding some of those things and seeing something and understanding something that nobody has ever seen and understood before. So why are there giant sea spiders in the Antarctic is a question I've grappled with in my career, and understanding that is one of the biggest kicks I've ever got. You can't, I can't get better than that. Seeing data that tells me the world doesn't work the way that we think it works is a massive kick for me. So I would say if you've got that, be a marine biologist, and that will give you the other things you need, because you need massive determination. You need to stick at it. There will be kickbacks. There'll be times when things don't go for you. There'll be rejections. When I write a paper, 9 out of 10 of my papers get rejected, and we have to rewrite them and we have to send them back. And sometimes the referees say, well, this is just rubbish. And you have to put up with the kickbacks. When we know they're not rubbish, we know the referees just disagree with our interpretation of what our data are saying. Then you rewrite it, you work out what the problem was, you solve that problem, and you go to it again. Grants, you only win 1 in 10 grants. So you spend a huge amount of time failing, and you have to learn to cope with failure. You have to stick at it. You have to not give up. And for me, the thing that's kept me going in that for a long time is because I just can't stop asking those questions. I can't visualize myself doing anything but asking questions about the way the world works and then trying to solve those questions.
Thank you for answering that one honestly, because I think that's really key. And we even had a question coming from Healing Academy. Um, Someone called Sam from their class wanted to ask about the salary of a polar marine biologist. So it's nice to be frank up there and say that it's not the best, most well-paid job in the world, but you do it for the science.
You
So
do get to go to some nice places too. So part of my work has taken me not just to the Antarctic, but we look at how animals are different. So we do comparisons. So I've worked in Fiordland in New Zealand several times, the fjords in Chile. I've worked in Stewart Island in New Zealand. I've worked in Australia. I've gone to the Barrier Reef, and my research has taken me there, and I've won grants to go and do work there. I've dived in California. I've dived in the Arctic. I've dived in Scotland. I've dived in the Mediterranean. I've dived all those places because the research questions have taken me there, and the grant funding has taken me to answer those questions. So there are some benefits. You'll get paid twice as much as an accountant or a lawyer or a doctor or any of the well-known professions.
very, very interesting indeed. We have a final formal question before we move on to just pure student questions, which is, what are your key pieces of advice that you would give to someone who wants to go into this career?
I would say try and get experience of the marine environment. If you're close enough to a sea life centre, volunteer for a sea life centre. go and get summer jobs in a sea life centre. If you're close enough to a university that does marine biology, Plymouth, Southampton, Brighton, Hull, Swansea, Bangor, Newcastle, any of those, look for volunteering opportunities to go and find out what people do. Then, if you can, as you progress, volunteer for some of the expeditions. I mentioned Wallacea. There is a whole suite of them out there that take volunteers, and the volunteers help them with pieces of biodiversity research in the marine environment. Any experience you can get is really valuable. For me, learning to scuba dive, learning to drive a small boat, they give you opportunities that you wouldn't otherwise get. And they allow you to get career opportunities because you've got expertise that allows you to do the practical things that are needed. And even if, as a younger person, you've not got the experience and the expertise to do the science, if you can help people drive boats around and you can help them with their scuba diving, you can then get progressively more involved through maybe your scuba club if it's at one of those universities. So I would say do well in your A levels, learn to scuba dive, learn to drive boats, go to a university that does marine biology, get involved in the extra activities at those universities, get involved with the diving club, go on expeditions with the diving club, and then volunteer to help with the research programs at those universities. Those things always help you progress through a career.
Awesome. The really good piece of advice that, and we've had a few questions talk about experience and things like that. So good for mentioning that and getting that in. Our next question, though, I really like this one. It comes from a CWA school, and it's, what's the most exciting investigation that you have been a part of in your life so far?
There's too many. At the moment, I'm really excited by the work that we're doing on viscosity of cells. So, as I mentioned in the talk, as you cool things down, the environment inside the cell changes. And we've been measuring the viscosity, the stickiness of cytoplasm in fish. And the stickiness in the cytoplasm of our Antarctic fish is maybe twice as much as it is in fish living around the UK. So that's almost like trying to make things work through treacle instead of water. And if you think about trying to move things around your cell, and cells do this, they move their proteins around, they move resources around, they move even the organelles inside the cell, even things like mitochondria move about the cell. Well, if the resistance to that is twice as high, then that's a big problem. So at the moment, I'm getting a huge amount of excitement from a PhD student and a master's student we've got working on that viscosity problem. And how do we measure that? And how do we measure it well? And how do we measure the effects in the cells? So I've got a PhD student who has built a light microscope that works at 0 degrees. And it's the first time anybody's ever done this anywhere, so that you can have living cells underneath the microscope at 0 degrees, and you can watch what happens inside the cells. And then we can change the environment a little bit and we can see what effect it has. Nobody's ever done that before. I'm really excited by that. But I was also really excited when we went and worked out that you get giant sea spiders because there's more oxygen in the Antarctic marine environment, twice as much as in the tropics, and that these animals are using it 20 times slower than similar animals elsewhere. So if you reduce the costs down and there's more resource, you can get a lot bigger. But then the trade-off with that is they grow really slowly. So these things have to grow for 10 times as long to get to a bigger size than they do elsewhere, because they grow slowly, but the cap on the size they can get to is raised and raised and raised. So the consequences of this, I just get really excited about that.
I love it. I wanted to get as many questions in. I'm going to keep firing them at you. So Copley Academy have asked, what's the most challenging part of being a marine biologist?
The knockbacks, the fact that you can see something that's really important. So, for instance, we can see that the environment is changing really fast on the Antarctic Peninsula where I work. The warming in the end of the last century for 50 years on the peninsula was as fast as anywhere on Earth, as fast as the Arctic. It looks like sea ice. In fact, the last 5 years, sea ice has started to decline rapidly around the Antarctic and especially on the peninsula. We know this is changing the biodiversity. We tried 2-3 times a year to win grants to go and measure the changes in that biodiversity on the Antarctic Peninsula, and they've all failed. And I find that massively difficult to cope with. It's the kickbacks. It's not being able to spend our time and my group's time going and doing the research, having to win the money to get the research. They're not being able to win the money to get the research. I spend 60 to 70% of my time trying to get money for us to do research instead of doing the research. I find that really difficult to cope with.
Yeah, definitely difficult thing to have to deal with, I guess, all that change. It must be very like all hands on deck out there.
It is.
Our next question comes from Hereford Sixth Form College, and they've asked, "What is your favourite topics in the field of this work?"
I'm really a mechanistic biologist. So there are marine biologists who are interested in patterns. So they look at what lives where, and they look at the patterns of change from place to place. There are biologists who look at why those patterns change. So the ecology, and they're looking at how temperature affects those patterns, or how the ability to disperse. So for instance, how long an animal might have floating larvae in the water and allowing them to colonize new places. As you'll have gathered from some of my previous questions, I'm interested in how the mechanisms are different. How is it that fish can live without red blood cells? There are 16 species of fish in the Antarctic that don't have red blood cells. You, me, every other animal with a backbone, all the other fish, all the other hundreds, thousands of species of fish, all the amphibians, all the reptiles, they carry 95 to 98% of their oxygen in red blood cells. And we've got 16 species of fish in the Antarctic that don't have red blood cells. And I'm really interested in the mechanisms of how they carry their oxygen. How do they get enough oxygen to their muscles to get the muscles to work? How do they get enough oxygen in to allow them to chase food and eat food and then process that food? How have their gills changed? How has their circulation system changed? And then I like asking questions about, well, when they went from having red blood cells to not having red blood cells, what changed first? And what has followed on from those changes? So I'm all about the mechanisms. And it's why I say to you, those fish are more different than any other animal with a backbone on the planet, because they're the only ones that don't have red blood cells. And yet I don't see anybody having a massive campaign to save the Antarctic ice fish. And for me, I find that really, well, sad in one sense, but also something that I have to tell the world about.
Which you're helping to do today, in a way. Our next question, we have time for just a couple more, comes from Libby from Healing Academy, and she has asked, what's your favourite animal?
So there are lots of answers to this question. I had an experience with meerkats 3 or 4 years ago where we went and sat in the meerkat enclosure and they climbed all over me, and I found that absolutely a real turn on for an experience. However, I have lots of animals in the sea that I am just absolutely blown away by. We have a 40-armed starfish in the Antarctic that grows 75 centimeters across, and I find it really hard not to just go and spend time looking at those when there's one on a dive that I'm on. I got into brachiopods, which are one of the longest lived animals on Earth. So they've been here for 500 million years. They're one of the few groups of animals that have been around for 500 million years. They have the lowest metabolic rates of anything. You find it hard to measure their oxygen consumption and their respiration rates because they breathe so little. If you look at one, they are 95% skeleton. So they are only 5% soft tissue. And we are orders of magnitude more than that. And I'm intellectually absolutely fascinated by how they have been so successful over such a long period of time, and yet appear to be just a smear of living thing on the outside of a skeleton. I love giant sea spiders. I love huge jellyfish. We have jellyfish 2 meters across and 30 meters long in the Antarctic. And again, if one of those appears when we're in a dive, I just have to get in the water and look at it, because I'm blown away by how impressive and different the life is. Yeah, everybody else says whales, seals, penguins, things that are furry, feathers, big eyes. No, life isn't like that on Earth. The really impressive life is definitely invertebrate.
I like it. A very unusual answer to a great question there, Libby. So I think this might have to be our final question, and it's come a couple of times from different places. So Alder Carr High School have asked, what is the best A levels I should be looking at in order to get the best chance of doing this career?
So there's lots of routes. There are people who work more on the mathematical side and are modelers doing AI. They tend not to do the field marine biology, but they do the analyses, and they do have a career, if you like, more on the computing side. We're doing some field work, and for them, you'll need maths. You'll need maths, maybe some physics. If you want to be a field marine biologist, which is what most people want to be, if you're true, and most of the students that I talk to as undergraduates who want to be marine biologists, they want to work on seals, they want to work on dolphins, they want to work on penguins. They don't want to work on what I think of as marine biology. Then you'll need a mix of biology, chemistry, and physics, and those are the three ones that will get you into a marine biology degree. And if you like, that's the step you have to think about. If you are interested, look at a course as you go through your A levels, look at the university courses, look at the requirements, look at what they do, look at what will set you up best for the specific degree you want to do, and do those A levels. But look at them before you start your A levels. Try and work in advance. What do I need for a marine biology degree? And then once you're in your A levels, look for what you'll need after your degree for your master's courses, and look in advance. Don't just let it happen.
Love that. I really like that as a nice way to end the session on. So as we look to kind of wrap up today's session, Lloyd, do you have any final thoughts and feelings that you'd like to leave with our young people as we say goodbye?
So the world is now changing faster than it has changed possibly in the history of the Earth, outside of mass extinctions and bolide impacts or meteorite impacts. Biodiversity is changing really fast, and it's going to change like this for the next 20, 30, 40 years. I think it's possibly the most exciting time to be a marine biologist, because it's depressing because we're killing things off, but also getting understanding of what's happening, trying to change that, trying to slow it down, trying to mitigate it, trying to do the conservation that's going to save some species. I think those are some of the most important things that can be done on Earth. And they're the careers that students that are in the 14 to 18-year-old age groups now, you're going to be doing that in 10 years time and 20 years time. And I wish I was a marine biologist now, because coming up through the age groups, because there's so much that needs to be done. So I think it's depressing because we're doing really bad things. We can contribute as marine biologists to giving the evidence to try and get the rest of society to slow down. And that, I think, is as big a job as anybody can do anywhere.